Welcome to G Fun Facts Online. The clinking of glasses has echoed through human history for millennia, a universal sound of celebration, ritual, and relaxation. For thousands of years, the contents of those glasses were dictated by the caprices of wild yeast and the preservative necessity of alcohol. Fermentation was a mystical gift, a way to purify water and store the caloric energy of grain and fruit. But in the 21st century, the script is being rewritten. We are witnessing the dawn of a new epoch in beverage science, a renaissance driven not by the need for intoxication, but by the desire for flavor, health, and mastery over the very molecules that define what we drink. This is the era of modern zymology and dealcoholization, and we are going to explore exactly how scientists are creating wines that never saw a grape, and beers that offer the complex bouquet of a double IPA without a single drop of alcohol. Zymology, the science of fermentation, was formally christened in the mid-19th century when Louis Pasteur peered through his microscope and connected the life without air of yeast cells to the conversion of sugar into alcohol. For over a century, the goal of zymology was singularity, maximizing alcohol yield, ensuring consistent fermentation, and breeding traditional yeast, known as Saccharomyces cerevisiae, to be more robust and alcohol-tolerant. Traditional zymology was an exercise in more. Modern zymology, however, is an exercise in less. It is the science of subtraction and precise molecular modulation. The modern zymologist is no longer just a shepherd of yeast. They are a metabolic engineer. As consumer habits shift toward wellness, the industry faced an existential crisis. How do you remove the defining ingredient of your product without destroying its soul? The answer lies in two distinct paths: biological attenuation, which stops the alcohol before it starts, and physical dealcoholization, which removes the alcohol after it is made. Let us start with the biology and the rise of the lazy yeasts. For decades, a brewer's worst nightmare was a stuck fermentation, a batch where the yeast stopped eating sugar before the job was done, leaving a sweet, unpalatable mess. Today, that nightmare is a feature. Modern zymology has turned its attention to maltose-negative yeasts. In a standard unfermented beer, known as wort, the sugar profile is roughly 10 to 15% simple glucose and 50 to 60% complex maltose. Traditional brewer's yeast is a glutton. It devours the glucose first, then switches on specific genes in the MAL locus to transport and break down maltose, converting almost everything into ethanol and carbon dioxide. But what if you used a yeast that was genetically incapable of eating maltose? Enter strains like Saccharomyces ludwigii and Torulaspora delbruci. These are the new stars of the non-alcoholic brewing world. They are lazy by design. The primary difference lies in the cell membrane. In standard yeast, a protein channel called the AGT1 permease acts like a gatekeeper, ushering maltose into the cell. In maltose-negative strains, the gene encoding this permease is absent or mutated. The yeast literally cannot see the maltose. It eats the low-hanging fruit of the glucose, produces a tiny amount of alcohol, usually under 0.5%, and a burst of flavor compounds, and then goes dormant. This biological approach, known as arrested fermentation, is far superior to simply boiling beer to remove alcohol, which results in a cooked, cardboard-like taste. However, arrested fermentation comes with a challenge. Because the maltose is left behind, the resulting beverage can taste unpleasantly sweet and grain-like. This is where the artistry of modern zymology comes in. Brewers like Athletic Brewing Company use a multi-hurdle approach. They mash their grains at higher temperatures, like 76 degrees Celsius, favoring an enzyme called alpha amylase. This creates long-chain unfermentable sugars called dextrins instead of maltose, adding body without the cloying sweetness. They also pair these lazy yeasts with specific hop profiles and use non-conventional yeasts like Mechnikovia pulcherrima, found on fruits and flowers, which produces distinct floral notes to trick the brain into perceiving a fermented complexity. They might even use Pichia cluiveri, a yeast that produces massive amounts of tropical fruit aromas, to distract the palate. But biological methods have limits. You cannot easily make a dry, full-bodied Cabernet Sauvignon using a yeast that refuses to ferment sugar. For wine, and for fully fermented zero-alcohol beers like Heineken 0.0, we must turn to the brute force of physics. Dealcoholization is the process of taking a fully fermented beverage and chemically dissecting it. The goal is to remove the ethanol molecule while leaving behind the thousands of other molecules that contribute to flavor, aroma, texture, and color. This is incredibly difficult because ethanol is a solvent that holds aromatic compounds in solution. When you rip the alcohol out, the aromas tend to cling to it and leave too. There are three primary technologies dominating this field today. The first is vacuum distillation. We all learn in high school physics that the boiling point of a liquid depends on the surrounding pressure. At sea level, ethanol boils at around 78 degrees Celsius. Boiling wine at 78 degrees destroys it, cooking off delicate esters and terpenes. Vacuum distillation solves this by lowering the atmospheric pressure inside the vessel. Under a strong vacuum, the boiling point of ethanol drops dramatically to as low as 28 to 30 degrees Celsius. The wine gently cascades down through a series of plates, while the vacuum pulls the volatile ethanol vapors out, never getting hot enough to cook. However, even at 30 degrees, the most volatile aroma compounds are eager to evaporate. To combat this aroma loss, engineers use aroma traps to catch these fleeing essence molecules and add them back later. The second technology is the spinning cone column, widely considered the gold standard. Picture a gleaming vertical steel cylinder that looks like a rocket ship component. Inside is a central rotating shaft fitted with dozens of inverted metal cones, interleaved with stationary cones attached to the column wall. Wine is fed into the top under vacuum. As it hits the first spinning cone, centrifugal force spins the liquid out into a film thinner than a piece of paper. Simultaneously, cool stripping steam rises from the bottom. The massive surface area allows volatile molecules to be stripped out with incredible efficiency. This process is usually performed in two passes. Pass 1 is run gently to strip out only the ultra-volatile aroma compounds, producing a highly concentrated aroma essence. Pass 2 is run aggressively to strip out the ethanol. Finally, the aroma essence from Pass 1 is blended back into the dealcoholized base, retaining the fresh character of the original grape. The third technology is membrane filtration, specifically reverse osmosis and nanofiltration. While the spinning cone uses centrifugal force and steam, reverse osmosis uses pressure and porosity. Wine is pumped at high pressure, often 30 to 70 times normal atmospheric pressure, against a semi-permeable membrane. The pores in this membrane are infinitesimally small, less than 1 nanometer, with a molecular weight cutoff of about 100 daltons. These pores block large molecules like tannins, color compounds, and complex sugars. However, water and ethanol are tiny molecules, small enough to be squeezed through. On one side, the retentate, you get a concentrated syrup of wine flavor. On the other side, the permeate, you get a clear mixture of water and alcohol. The alcohol is distilled away, and the water is fed back into the loop. Nanofiltration works similarly but with slightly larger pores, allowing it to fine-tune the sugar and acid profile. But removing alcohol is only half the battle. Putting the sensation of alcohol back in is arguably harder. Ethanol provides body and viscosity, perceived sweetness, the classic trigeminal burn in the throat, and volatility to lift aromas to your nose. Removing it leaves a thin, acidic, watery beverage. Modern zymology addresses this through reconstruction. To replace the viscosity, producers use natural hydrocolloids, glycerol, and manoproteins derived from yeast cell walls to restore a creamy mouthfeel. To simulate the burn, the industry uses botanical extracts, like Szechuan pepper, which contains hydroxy-alpha-sanshool for a tingling sensation, or grains of paradise for a peppery warmth. Biotech firms are even developing blends that target the exact same transient receptor potential channels in the throat that ethanol activates, creating a phantom burn. To compensate for lost aroma volatility, zymologists over-index on aromatic yeasts during fermentation, creating 10 times the normal amount of banana, pear, or apple esters, so enough aroma survives the dealcoholization process. Looking beyond subtraction, the future of the industry lies in molecular assembly and synthetic spirits. Companies like Endless West are challenging the definition of wine by analyzing the molecular fingerprint of a target beverage, identifying the exact ratio of acids, esters, and tannins that make a specific vintage taste the way it does. Then, they build it from scratch, sourcing molecules from more efficient plants like lemon peels, instead of fermenting grapes. You do not have to remove the alcohol because you simply never put it in. This avoids the trauma of dealcoholization entirely. Another frontier is precision fermentation, genetically modifying microbes to produce specific complex molecules like valencine for orange aromas or linalool for floral notes instead of ethanol. This enables function stacking, creating beverages brewed with yeast that produce B vitamins for recovery or infused with L-theanine and ashwagandha for relaxation without intoxication. This technological leap also brings significant environmental benefits. Traditional thermal dealcoholization is energy-intensive, increasing a beverage's carbon footprint by up to 30% and generating massive amounts of wastewater. The biological approach using maltose-negative yeast is much more sustainable, requiring no extra energy steps. However, the molecular assembly approach claims the ultimate sustainability crown. By skipping agriculture entirely, it reduces water use by 90% and carbon emissions by over 80%. As climate change threatens traditional vineyards, lab-grown non-alcoholic wine becomes a necessity. Navigating this new world also means navigating a global regulatory mosaic. In the United States, non-alcoholic means under 0.5% alcohol by volume, while alcohol-free means 0.0%. The European Union generally aligns with the 0.5% threshold, while Islamic halal markets demand 0.0% and strictly forbid products derived from alcoholic origins, effectively banning dealcoholized wines and pushing the market toward molecular assembly and arrested fermentation. These evolving regulations heavily dictate which scientific method a brand will deploy. Finally, all of this science poses a fascinating dilemma for the sommelier. A sommelier is trained to recognize terroir, the taste of the soil and climate. But removing alcohol alters the partition coefficient of aroma molecules. Fruit flavors become less volatile and hide in the liquid, while green, vegetative notes like pyrazines can become overpowering. Winemakers must act as flavor architects, harvesting grapes later or oak aging them before dealcoholization to ensure the right flavors survive the machine. We are currently surfing the third wave of non-alcoholic beverages. The first wave brought thin, punitive drinks. The second wave brought the craft revolution. Today, the third wave brings high-tech zymology, genetically tuned yeasts, spinning cone precision, membrane sieves, and molecular assembly have transformed the non-alcoholic aisle from a place of compromise to a place of pure innovation. We have proven that ethanol is not the only path to complexity or enjoyment. By manipulating biology and physics, we have learned to keep the ritual while changing the result. The glass is no longer half empty, it is full of science. Thank you for listening to this deep dive on G Fun Facts Online.